In this video, we will discuss about the CHIC2 knockout phenocopy, the STUB1 knockout in terms of the tumor response. So far, we have discussed about how these CD8 plus T cells with STUB1 knockout work against the tumor. So how this CHIC2 come up, that we will also discuss in the results that are presented in this figure. So let's start. So from the literature, the authors are able to know that this STUB1 plays a role towards E3 ubiquitations. So the hypothesis that STUB1 might be interacting with the target proteins and it leads to the ubiquitations of those proteins. So in order to check their hypothesis or validate their hypothesis, what they did is they did the mass spec analysis of the proteins that are ubiquitinated in the wild type CD8 plus cells and the STUB1 knockout CD8 plus cells. And what they're able to find out that these CHIC2 CHIC2 genes are been heavily down regulated in the case of this STUB1 which is present and when the STUB1 is knockout. So in the case where STUB1 is present, the CHIC2 is heavily down regulated. So from that they come up to a point that the CHIC2 might be interacting with this STUB1. So in order to validate that, what I did is they prepared a cell line where they electroporate these EL4 cells with this CHIC2 guide RNase, and these stable cell lines then they transduce this with V5 tagged CHICT, and also they transduce them with this CHIC with this IRF PD has been knocked down. So in this case, so they have now two different conditions. One which is this CHIC2 wild type, which has this V5 tag will interact with this STUB1, and from literatures also they are able to find out this IRF PD domain of this CHIC2 plays a role towards this STUB1 interactions. So they knocked down that part and checked whether CHIC2 will interact with the STUB1 or not, and then they compare these two different conditions using the immunoprecipitations of this V5. So in the case where wild type CHIC2 is injected, what they are able to see is that if the immunoprecipitations, they are able to see the immunoprecipitations of this STUB1, they are able to see this V5CH2 also. But in the case where delta IFRD domain is not been transduced, they are able to in the case of V5 immunoprecipitations, they are not able to see STUB1. So this indicates that this IRFPD plays a role towards its interaction with the STUB1, and also the STUB1 and CHIC2 interactions occurs in the wild type conditions. And here the whole cell lysate, in the whole cell lysate we can see that both this V5 CHIC2 and delta IFRD are present in the cells, and for the control they use vinzolane. Thereafter, once validating that there is interaction going on between STUB1 and CHIC2, they wanted to check their in vivo results. So this time they have the four different conditions. First one, they took these cell lines, that is OT1 naive CD8 plus T cells, and then they use four different conditions. First one is the control guide RNase, then the STUB1 knockout, CHIC2 knockout, and in the last case their STUB1 and CHIC2 both are knockout, and they mix them in the 25:25:25 and inject them into this B16 OVA tumor injected mice. And what they are able to find out that in terms of this IFN gamma, they check the IFN gamma readings. The IFN gamma fold change is high, around more than 1.5 times compared to the control in all the three cases. And for this IL-27, it also increases. So both of them are related to the activations of the CD8 plus T cells. And we can see that in all the three cases, and but the one thing that they're able to see that even in the dual knockout they do not see any significant increase, and it is comparable to the single knockout of STUB1 guide RNAs or CHIC2 guide RNAs, indicating that their dual knockout does not enhance its functions. But this STUB1 might be interacting with the CHIC2 in a way that is suppressing the CD8 plus response against the tumors. And when they check it about, now they wanted to check whether CHIC2 knockout can phenocopy the STUB1 knockout or not. So in that case, they use the STUB1 knockout and CHIC knockout and then check the CD8 plus T cells or the population that is transferred with the CH plus T cells, and they are able to see that the STUB1 results and CHIC2 results are similar in all the three cases that they have seen, that is SLAM F6 plus team 3 minus and SLAM F6 plus team 3 plus, SLAM F6 minus TM3 minus. And when they check it for this IFN gamma plus, here also they see the similar results for the STUB1 and CHIC2 knockout cells. And in terms of the exhaustion of the CD8 plus T cells, the results are similar for the STUB1 knockout and CHIC knockout. So this all indicates that the CHIC2 knockout phenocopy the STUB1 knockout in terms of the in CD8 plus T cells towards the this tumor response. Now they check the final actual outcomes, that is effect of these knockouts on the tumor volume, and for the control they can see the tumor volume increases significantly in a short duration of time, but for the all three cases they are not able to see the similar amount of delay in the tumor increase, indicating that the all three conditions has the similar effect on the CD8 plus T cells activations and its response against the tumor. That's all they have concluded in this figure, that CHIC2 knockout phenocopy the STUB1 knockout in terms of the tumor response in CD8 plus T cells. That's all for this video. If you need to understand more about these experiments in detail, you can go through the reference that has been provided in the description. Thank you for your time.